environmental issues that we're facing and potentially feel empowered to solve them.
That's great. I mean, we've spent 5 years now in this big STEM project where we're trying to get real science into schools, and we've succeeded in some things and failed in a bunch of things. But one of the things that's been really clear is that the connecting students directly with scientists is one of the best things we ever do. And yeah, so fantastic.
Yeah, I'm glad that you're able to be a part of it.
I'm glad for your moonshot.
Okay, so it's now 7 o'clock, so we'll go ahead and get started. Hello, everyone, from the Scientists in Every Florida School Initiative, a program led by the UF Thompson Earth System Institute. I am Brian Abramowitz, and along with my colleague, Stephanie Killingsworth, we would like to welcome you to our first Nature of Science webinar. Our mission at the Scientists in Every Florida School Initiative is to build long-term collaborative relationships between teachers and scientists, to better integrate current scientific research and big data into classroom lessons that adhere to Florida Sunshine State standards, and in doing so, to connect the scientist with every Florida school in the state. Today, we are joined by Dr. Rob Dunn. He is a William Neal Reynolds Distinguished Professor at NC State University. You may recognize him from his books Never Out of Season or Never Home Alone, or even from his over 150 peer-reviewed articles on ecology, biodiversity, and global change. Without further ado, I would like to turn it over to Dr. Rob Dunn.
Hi, folks. Thanks so much for the introduction. It's a pleasure to be here today. I'm going to try to do 3 things very quickly. Aware that I don't have infinite time. I'm used to like a 4-hour lecture period, and so I'm going to condense it all. First is to introduce you broadly to what we do in my lab. And so if you go to the Rob Dunn lab, you'll get a sense of what we do. And in general, we engage the public, both in formal and informal settings, in the process of science. And so let's see, if I screen share here, where was my magic button?
Should be right on the bottom of the green.
Yeah, I think I got it. What are you looking at? Is there a
Yeah,
You're
it's
still
your
seeing me?
sourdough science, never home alone.
Perfect. So we've done projects on sourdough bread, on the life in homes. We did a big project in which we had people wear different kinds of pants to see which life was living in their pants. We've done a big project on the life in shower heads, pollinators, belly button biodiversity. And in all of this work, we engage the public in doing science. And over the last 5 years, a lot of that work has happened in the classroom context. And so we're working through a project called Students Discover, and you should be able to see the link in your chat, to develop a series of curricular modules around these and other projects so you can take citizen science projects into the classroom. And so the idea here is that while meeting state and federal standards, that students can be part of real scientific projects where they're contributing data to those projects, and then they can ultimately see where their data fit into the bigger picture. And so we have projects on cadavers, which have been really interesting, actually. It turns out that although students dissect animals all around the world every year, for the most part, no data are compiled on those dissections. And so we developed a project where students can help us figure out how different the bodies of different individuals of the same species are and what that means biologically. In the first version of that project, which we did with human cadavers in an undergraduate class, we documented about 50% more variation in intestine length than the first 26 people that were studied by the students than was known from the entire scientific literature before. And so through all these kinds of projects, we're seeing new sorts of discoveries. But there are two projects I want to focus on a little bit more today. And one is this project that's on the iNaturalist platform called Never Home Alone. And so if you go to iNaturalist and you type in Never Home Alone, you get to this project. And it's a project where we enlist the students to go around schools or homes to take pictures of the species they see around them every day. And the backstory in this project is several years ago, we became interested in what lived in homes with us. And when we talked to our colleagues, most of our colleagues thought it was the world's most boring thing we could ever study. But that in general to me is a sign that either it actually is boring or we're onto something fun. In this case, we were onto something fun. And so we decided to go into people's homes to study what we could find in those homes. And the first 50 homes we went into, we found more than 1,000 species of insects and their relatives. And which led us to the realization that there was much more going on in houses than we thought, which led us to develop this project on iNaturalist. And so as part of this project, what you can do is you sign up for iNaturalist, takes about 30 seconds, and then you just take pictures of what you find in your home. And so you go to iNaturalist, you go to this project, Never Home Alone, The Wildlife of Homes, and you can see if you click on observations, these are photos that students, families, and other folks have taken in their own houses of what they see. And so once you take a picture, people start to help you to identify it. And so, for example, this is an image of a false black widow that people have already agreed on the identification of. And it was just submitted, yesterday. And you can also see what other people around you have documented. And so we have a lot of samples from North Carolina. So if I type in North Carolina, you can see all these species. Of the species we find in North Carolina, these are the most common. As an interesting aside, this camel cricket, which is the most common thing people are seeing in their houses, when we first started studying it, was not even known to be indoors in North America. And that was just 4 years ago. And so it's the most common species in houses in North Carolina. It's the size of my thumb, which is sort of a normal human-sized thumb, and we were missing it. And so there's lots that kids can discover in houses, in their schools, that nobody really knows is there. And then there's some stuff we know pretty well. And so this is the first project, easy to engage in. We're developing curricular modules for it for both middle school and elementary school. And I'll tell you right now that I think biologically what's in Florida houses and schools is about as interesting as anything we'll find in the world. But we have very few contributions from Florida. And they tend to be on the herpetological side of things. And so if your kids contributed, they would be pretty, very useful and quite novel contributions to the overall project. And they don't have to be photos of living things. In fact, the best way to do this is to find light fixtures or windowsills in your school and go and check out what's living on them, or what's dead on them rather, and then to take pictures. If your school decides to do this, we can send you macro lenses for your iPhones or other phones, so that kids can take better pictures of these. How many people are online right now? About? Conceivably, if everybody I see is who's online, we could send all of you whole kits for your classroom to get all your kids taking pictures of what's in the school. And so this is one of the kinds of projects we do. But the other kind we do is one where we directly collaborate with teachers to develop a project from something more like scratch, where we're actually collaborating in the whole endeavor. And so this is a project on sourdough bread. And what I'm going to start with here is the mystery that we have. And the mystery is that we've studied, so I'll back up and say that sourdough bread, to make sourdough bread, you put flour out, bacteria and fungi colonize that flour, and they start to leaven it. They grow, they metabolize, the yeast the fungi produce carbon dioxide, the bacteria produce acidic flavors, and this is traditionally how all bread was made. We know very little about the biology of that process, and compared to most things, it's essentially gone unstudied. Well, when we were studying the life in houses in general, one of the things that we observed that was very unusual is that when we looked inside of houses, that we found many kinds of yeasts of the sort that we would expect to find in sourdough bread. And when we looked outside of houses, we saw many fewer. And so on the x-axis here, you see latitude. So 60 is far north. Where are you in Florida? Down here. And on the y-axis is the number of kinds of yeasts. And so what you can see is for a given latitude, the mean number of kinds of yeasts indoors is far more than outdoors. And so this is a really unusual observation, and we don't know what to make of it. But the trick is that the way we've studied these to date, we can't tell if these are living or dead. What we've done is to have people swab dust in their homes, and then we identify what's present in the dust based on the bits of DNA. But what we're now doing is to get teachers to work with their students to make sourdough starters both in the classroom and outdoors at the school, to try to see, is it easier to make a sourdough starter indoors than outdoors? Are there actually more useful yeasts indoors than outdoors? And have we somehow inadvertently cultivated a set of useful species indoors without realizing it over the last thousands of years? And so if this is a project that you're interested in being part of, if you make a sourdough starter in your class and outside your class and work with us to do it, you'll actually be part of the collaborative research paper we do on this question. And so you as a teacher will be an author on the paper, and your students in bulk, not by name, can be mentioned in the paper as part of this process. And so these are the kinds of things that we do, and we have many, many projects of varying ilks that are like these. And maybe I'll pause here to make sure that I'm being understood and make sure there aren't any questions. And Brian, you can tell me if I should be doing this differently here.
No, I think you're doing a great job.
Do people have questions about what I've said so far?
Just questions regarding how do you receive a lens if you want to be part of your project?
Well, depending on what's easiest for you, Brian, you can email you, and you can collate the emails and send them to me, or people can email me directly. It's probably a little easier if you collate, but either is fine.
Sure. What information would you need in that email?
Just an email and a school and maybe the age range of kids.
Okay.
And then we can also send along the curricular materials when they're finished.
Okay.
Any other questions?
I think that's it for now.
Oh, Bruce, did you have something? Oh, I think your mic's off.
I do. So are you also involved in world events or school events?
Yeah, so Andrea Lucky, who you have the good fortune now have down in Florida, started the School of Ants here at NC State when she was here, and then moved down to University of Florida, Gainesville, and brought that with her. And we now have a version of that called Ant Picnic that one can also do in the classroom. We historically had a version where people could send ants into us, which yielded all kinds of discoveries. But we no longer have the staff to have people identify all those ants. And so we have a new version that's more focused on identifying what ants are doing than which ants they are. But I can send you at a quiet moment, I'll post a link to the new version of that, Bruce, if that's useful.
Oh, I'm interested in having young scientists learn how to collect the ants, and I'm just interested in that project. And Andrea is a fellow of our institute, so I'm somewhat familiar with her work, but I've never connected with you before, and now I'm making that connection, so that's really interesting. Thank you.
Yeah, so if you go to, let's see if I will stop screen sharing, so Google for a second. Discover the ant life. Discoverants.com.
Okay, thanks.
That now has all of our existing resources for ant-based projects in the classroom.
Thanks.
And anybody else? Let's see. I think I can also post that somewhere here. Chat. Yes. So I'll with regard to the sourdough, one of the things that's been really intriguing is that when we look globally, so for the most part, when people study bread, they focus on industrial production of bread. And so how do you get the best yeast to make the most bread possible as fast as possible? And that's meant that things like sourdough bread have been much less well studied. And so we don't actually know, well, what governs why you have one kind of yeast, one place, one kind of bacteria, one place. And so we did a project where we engaged 1,000 people around the world to send us their sourdough starters. And one of the things that we found is that they were much, much more variable than anybody knew, and that people at home knew many more ways to make the sourdough starters than we had ever imagined. And so it's made us very aware that there's, especially in food-related science, that there's a lot of knowledge in homes and with families that we as scientists have really failed to engage around. And so looking to the future, one of the things we're really trying to do is to figure out what are new ways to do citizen science style projects in the classroom around, but at the same time to also engage families so that we get intergenerational knowledge being shared in families. So if there's a traditional way of making bread, of vinegar, of kimchi, of whatever sort of food it might be, that the family can come together around the science that's happening in the classroom. And so if anybody's interested in that kind of work too, we're also looking for teacher partners as we move forward on those sorts of projects.
What would be particularly helpful for you? You said that you don't know almost anything about insects in Florida houses, in Florida homes. what would you really like to see from schools to mobilize an understanding to sort of fill that void?
Well, I mean, if one could imagine, it's a great question, Bruce. Imagine we had 50 schools in which we had a good set of photos of the insects found in the light fixtures and the windowsills in those schools. I suspect we would almost immediately see new species of insects, new to science, not just new to houses. And we would get a much better sense of what's common and what's not common in Florida. And so even having a relatively small number of schools start to gather those kinds of data, I think would be a big help. And I think because Florida, in terms of climate, is really Florida has a climate that many more of us are going to share in the future. For most models, North Carolina's future climate is the sort of Miami's current climate. Not too distant future. It's supposed to be 96 here on Thursday.
Whoa.
And so understanding which species are going to move north as climate changes assumes that we have some sense of which species are there already. And the species that are going to most directly affect us are the species that are most immediate to us. And so if we're going to know about any of them, it seems like it behooves us to know what's going on with those species in homes. And so I think in that way, Florida is actually, the data will be more interesting and more valuable than almost any place else we could look in the US.
That was actually something that you just hit on. I know in talking with Andrea, she was mentioning some migratory species that were tropical and they're kind of moving subtropically now because of climate and some of the changes. So that just hit the nail on the head for me, kind of a revelation that we're kind of that ground zero, if you will.
Yeah, I think, for sure. And there are lots of species that are kind of waiting out in Florida right now. That every time you get a frost, they die back. But each year that there are fewer frosts, they inch a little bit farther north. And what Andrea will be quick to say is that for many of those, nobody's studying them at all. And so spiders are a good example of this. For most of the spiders we find in homes, we can't find an expert who can tell us, well, are you sure that this is what this is? And so that speaks to how little we know about many of these species in general. And some are quite large. And so it may be that there's something enormous in your school that you assume everybody else knows what it is, but it's totally new. And so I think both it has an important component in terms of what we can offer to applied science. And, if you're the school that finds a totally new species to Florida, that's a pretty fun day. And so I think that part's exciting. The other part we've started to do too is to start to think about, well, if these species are immediate to us, how can we make their values more apparent to students? And so we've started to do a series of projects where we hone in on that. And so, for example, to go back to Bruce's question, one of the School of Ants things we did was we invited a group of teachers to have their students send us ants that they thought might be particularly likely to have bacteria or fungi on them that would be able to break down fats. And so we gave them some background on why we think those should be on ants in general. Ants, and we looked in those ants, and in looking at those ants collected from school kids in North Carolina, we found out the fungus that's able to break down fats faster than any fungus so far known from the literature, from a backyard ant from a North Carolina school. And Novozymes is now interested in that because breaking down fats is what they need to do to help clean your clothes and lots of other things. And so I think starting to be able to remind students that there's this life around them we don't know about, that knowing about it is important, that what they see can be useful, that pulling those threads all the way through, I think can be really powerful. And I think in Florida, I'm pandering, but I think it's the best place in the US to do it.
Excellent.
So are there other citizen science projects that primarily focus on Florida and K-12 engagement that you know of? Is there, I know that there must be some citizenscience.org or some other clearinghouses
Yeah.
for projects, but I'm just wondering whether this is already being done in Florida and we just don't know about it in our project, or is it really breaking new ground, and which direction should we go? So I think that's two questions. One is, how do we find out what's already being done in Florida? And how would we know what the scientists would want to know if we were to mobilize classrooms throughout the state to help with a citizen science initiative?
Yeah, those are. I will try to offer pithy questions to those big fun, pithy answers to those big fun questions. The first one is SciStarter.com is the best U.S. clearinghouse for outdoor citizen science projects, and they have now a variety of new resources for schools. Some work better than others, but they're there and they're a great resource, and they care a lot about schools. And they actually have there's one Florida county where SciStarter has a bigger footprint, and I'm going to forget what it is now, but I can look it up. The second question, are there existing Florida projects that you should particularly focus on? And I guess I don't know right off the bat. Yuri Hulser, who's at University of Florida, has a bark beetle project it's not Florida focused, but he's in Florida, and so it's immediate to his interests. I can look some more offline, Bruce, but I think a lot of citizen science projects are now pretty regional or global. So I'm not thinking of them off the bat for Florida per se, but it is worth looking. I think one of the things we've had success with, and is thinking about, is there a citizen science project we can focus on this year and do something big and statewide, or we do it at the university, so campus wide, and then another one we might focus on the next year. And so one way to do that is to find university scientists who are particularly interested in a particular project, have them be a key partner who can really engage schools for a particular year, and then you shift in the subsequent year. And we've had success with that. If that's of interest. Because I think, there's so many scientist resources in Florida, it would be a shame not to capitalize on what people want to know there, I guess, which is part of the nature of your question too. Am I answering what you were interested in there? Yeah.
Oh yeah. I'm just trying to think about how we can begin to mobilize some projects focused around scientists' needs that could be fulfilled only if they had citizen science projects that were involving schools. I have a friend who's a water chemist, and he said that on Earth Day next year, he'd like to have 500 schools throughout Florida just go to their local lakes or streams or near the oceans, and he was going to send them plastic containers, and he just wanted them to collect a plastic container of water all at the same time on the same day, on Earth Day. And then he had an idea about how he'd want to use those. So that's a really easy thing to do, and it's sort of a team-based approach. And that's just one example. He was more interested, frankly, in the fresh water, but and then sort of some of the estuarine areas as well. But I'm just interested in how we can generate more of engagement from Florida schools. I also, the yeast thing for me is interesting, but I'm wondering, do kids find yeast interesting? I think that, like, charismatic, ants to me are fascinating, and butterflies, I think kids naturally associate with those. I'm just wondering whether, how do you make the yeast exciting to young citizen scientists?
Well, they're stinky, right? So that's always appealing in the classroom.
Okay, that would be good. So that's all you need to say is that they're stinky.
Yeah, and you get to eat the bread, and so I think
Okay.
a key component of those is the sort of revelatory nature of realizing that food comes from somewhere, that you can make food, that it's biological. That once you have a stinky jar of stuff in the classroom growing, and you might be eating that later, that draws kids in. But then once you realize that stinky jar ultimately yields something that's absolutely delicious, it's a different kind of project, and but we've had very good success with it in terms of kids being engaged. And I would say too that my experience is that the best thing is a range of kinds of projects which are going to get different kids. It may be the kids that want to go catch an ant are not the same kids that find the bread mysterious. But if you have this range of projects, you're getting these different kids interested for different reasons. The other project that would be any project that capitalizes on the biodiversity of Florida, I think is really well suited to Florida. And so another one would be e-mammal, and e-mammal and candid critters. And so this is a project to get camera traps out at schools so kids can document which bigger things are around schools. And in North Carolina, it's sometimes boring because sometimes you see the butt of a squirrel like 100 times and that's what you get. But in Florida, there are quite a few species that you could see by schools that are quite rare and not well documented. And so that might be a fun one. My experience is that when you ask scientists when you start from scratch with scientists about what you might do in classrooms, is that your water example is sort of the opposite of my general experience, which is that the water example is very clean, it's very straightforward, you can see what you would do. That what scientists very often imagine is multi-partite and it's going to take 7 weeks in the classroom. And so that's one of the challenges we find, because we make many projects, and so we think a lot about how do you best bring a scientist and a teacher together and make something that will work. And that's a frequent challenge, is that how do you make something that's small enough to happen in a day in the classroom or maybe 2 days, while also having something that appeals to the scientists.
Dr. Dunn, I really love the opportunities that you've spoken about and how schools and teachers can get involved. But I think going forward, one of the biggest things district leaders, teachers, instructional coaches would ask at this point is, what does this have to do with learning standards? So that being said, Stephanie and I came up with a few to potentially talk about how we can relate this back to classroom instruction and the Florida Sunshine Learning Standards. I'm not sure, Stephanie, if you'd like to speak a little bit about those. You're on mute.
Hold on, unmute. There we go.
There you go.
I was going to say I'm going to share my screen, as I'm sure you know now. And let's see here. I've lost part of my title, but that's okay. We threw up a couple of standards by elementary, middle, and high school that follow the nature of science that we felt would be shoo-ins for what Dr. Dunn has been speaking about today with regard to citizen science projects, and also understanding that science doesn't always fit the formal experimentation mode for thinking. And then also that there's often times where it's a collaborative effect where you have different entities in different fields of science working together to solve a problem or ask questions. So we threw these up from an elementary, middle, and high school standard for you guys to look at and kind of think about and even ponder what other standards within the curriculum do you see some of the stuff that you've heard tonight fitting? Where can some of these benchmarks maybe be hit? And if you want, you can unmute yourself and chime in if you'd like. This document, by the way, is also in the group chat, and I can modify it if we need to. Anybody want to add anything to that doc?
Monica is typing. I think any standard will fit. I think that's what, is that what you're saying, Monica?
Either any or no.
Looks like any. All the nature of science ones. You can hit a lot of them, right? Like observations.
Yeah.
Yeah. Drawing inferences, maybe theory. You could certainly talk about maybe creating models or collecting big data sets, analyzing data, maybe even graphing things with students. If you've got a series of data points for various species, for example, on the Never Home Alone example that was given, those things might be something that you could play with in class as far as data. What else? Are there any standards that are more content-based? Life sciences, for example, since some of these had a pretty good connection to life sciences. Anything for, I see someone, the 1.6, Nature of Science 1.6 as well.
This is great. What a great idea.
So I will put in,
I'll also say for projects like this, if anybody ever wanted any data, any other kind of thing from a project to build it into curricular materials or to collaborate on something, we're always game for that.
where is that right here? 1.6, that'd be great.
I saw Monica just also added the opportunity for cross-curricular or multidisciplinary between reading, writing, and speaking standards as well.
Ah, that's a good point. Here, I'll just leave the 1.6 so that I can actually find my screen again. I've lost it. Awesome. Good, so you're seeing some that you can tie, Monica? And 1.5 too. Excellent. So as you all think about that, this document along with all of the links are posted in the group chat, and we've also recorded this video tonight, so it will be posted on our website, and you have access to it if there's anything that you want to pull or go back and kind of review or reflect upon, you can do so. And I think maybe these last several minutes, if we can, we'd like to hear from teachers. Oh, there's another message, let me get that too. But we'd like to hear from teachers to find out what other topics are you interested in hearing from? What sorts of things would you like us to target benchmark-wise in future webinars so that we can maximize these events for you? This is a bioscience article, I think. I'm going to stop sharing the screen here and I'll copy this and paste it in there. Excellent. Mammal data. You got it. Okay. I'm going to unmute you guys so that you all can chime in. Anything?
Hi, this is Christy.
Hi, Christy.
From Seminole County. I was thinking of specifically when you're talking about the yeast, you know, 7th grade is, it's the big heavy hitter of the L15s and the L14s with learning the makeup of the cell.
Yes.
So comparing the fungal cell to plant and animal cells. And then also the 6 L's are, you know, diversity and not really evolution, but the differences in living things. And then in elementary school, they hit plant and animal structures pretty hard too. So that can take you down into the kids taking pictures of arthropods and stuff around school and around home.
Excellent.
Is this a okay time to ask a couple questions?
Yes, please.
Okay. The first thing is, if it is possible to do so that that's accessible, because we kind of flip through a lot of things fairly quickly. That'd probably be helpful to me. Plus, I could share that with some of my colleagues. But I'm also kind of interested in the 2 projects that Dr. Dunn brought up, the using the macro lens to record insects. I've got a lot of science fair projects going, and I'll probably have a few more, and that might fit into that. Also, I actually have been running yeast type projects for a number of years as science fair projects. In fact, I got one going right now. I'd like to have some more information on. We've just been using store-bought yeast, but if I could get some good information on making starter from yeast that's collected in buildings or outside, I would be interested in that.
Stephanie, can you maybe collect these questions and I can respond to them all in a Google Doc with links and good resources?
Yes, I will.
Great. Yeah, thank you for those questions. Rather than try to squeeze everything in right now.
About the level of student interest in yeast. When yeast is brewing, it smells like beer, and a lot of students are familiar with that smell. So that has a pretty high interest to them when it's going on in the classroom because of that. And they also like to see the fact that it's foaming up when you're making it go pretty fast using sugar. So I think it's pretty high interest activity.
But for those, so in North Carolina, our rules about what we can grow in the classroom are pretty strict. And so some kinds of projects that would be fun we can't do, or we can only do in some schools. But you know, a lot of the microbes that we rely on for key societal needs come from insects in the first place. And so there are a lot of fun science fair projects that are safe that you could do, like finding new yeast from insects for making bread or beer. I guess beer not in the U.S. model, but bread, let's say. We think that most of the vinegar bacteria originally come from carpenter ants. There are a bunch of really straightforward questions in that vein that nobody's addressed. And so in the context of science fairs, if anybody ever wants a list of 72 things scientists think are interesting but haven't gotten around to, we can send that too.
Now that I actually have some carpenter ants in my yard, so that might be a possibility.
And Richard, I want to make sure you're aware there's a chat box, and I'm putting everything in that chat box, but I'll also be creating a doc and I can send it off to folks if you'd like to. And this is going to be on the website.
Okay.
Are there any additional questions?
Thank you for doing this. This is fantastic.
Yes, thank you so much.
Do you have a website that has a lot of your projects listed on it?
It's in the chat.
Okay.
Yep. Excellent.
I'd
Thank
like
you,
to thank
Dr.
you
Dunn,
all.
so much.
Yeah,
Yes, I'd like to thank you all for being present at our first webinar. I'd also like to extend our gratitude one more time to Dr. Dunn. I really appreciate his time and explaining his research to all of us here.